In 1992, evolutionary biologist Keith Stewart Thompson wrote, "A matter of unfinished business for biologists is the identification of evolution's smoking gun. The smoking gun of evolution is speciation, not local adaptation and differentiation of populations." In 1996, biologists Scott Gilbert, John Opitz, and Rudolf Raff wrote, "The origin of species, Darwin's problem, remains unsolved. Speciation, the process by which one species splits into two or more species, is the crux of biological evolution. Yet, despite the leaps and bounds made in biology in 160 years, this enigma remains unresolved for Darwin." Take, for instance, bacteriology. Bacteria, with their short generation times and large populations, are the perfect specimens for this kind of study. Yet, even after 150 years of bacterial research, there's no definitive evidence of one species of bacteria morphing into another. This lingering question, this unfinished business, is not due to a lack of effort or intellect, but rather the complexity of the subject itself. The puzzle of speciation is a formidable one, challenging our understanding of life and its many faces. As Thompson concluded, the identification of evolution's smoking gun, speciation, is a matter of unfinished business. The most promising line of investigation for speciation was Richard Lenski's long-term evolution experiment. In 1988, Professor Lenski at Michigan State University started a fascinating long-term evolution experiment on E. coli. This experiment was a landmark study in the field of evolutionary biology, as it sought to observe and document the process of evolution in real time. The experiment involved 12 identical bacterial populations, all derived from the same E. coli ancestor. These populations were cultured in separate flasks, and every day a portion of each population was transferred to a fresh flask with a glucose-limited medium. This process was repeated for over 20 years, equating to more than 60,000 bacterial generations. In 2008, a significant development occurred. One of these 12 bacterial lines developed a new trait. It was able to use citrate, a compound in the solution, as a source of food. This was a major departure from the ancestral E. coli strain, which could not utilize citrate in the same way. Lenski believed this to be a new species, marking a potential smoking gun of evolution. In 2014, Lenski specifically claimed that he finally had proof of speciation. He stated, "I would argue that citrate users are, or are becoming, a new species." However, this claim from Lenski was not without its critics. Controversy arose, with many questioning whether this was indeed a case of speciation or simply an adaptation within the same species. The debate was far from over. Enter Scott Minnick and his team, who found Lenski's claim for observed speciation to be overblown. Minnick questioned the interpretation of Lenski's findings in a 2016 paper titled "Rapid Evolution of Citrate Utilization by Escherichia coli by Direct Selection." Minnick's argument hinged on the belief that E. coli cannot use citrate aerobically. In his own long-term evolution experiments, he found a single aerobic citrate-utilizing E. coli strain after 33,000 generations, roughly 15 years. This was interpreted as a speciation event, yet Minnick and his team believed it was not. They conducted similar experiments and isolated 46 independent citrate-utilizing mutants in as few as 12 to 100 generations. Michael B., another prominent figure in the field, weighed in on Minnick's experiment. He commented, "The bottom line is that they were able to repeatedly isolate the same mutants Lenski's lab did as easily as falling off a log, within weeks, not decades." In a 2020 article titled "Citrate Death Spiral," Behe further declared, "Let me emphasize, the only result from the decades-long 50,000-plus generation E. coli evolution experiment that even seemed at first blush like it had a bit of potential to yield a novel pathway in the bacterium has resulted instead in spectacular devolution." So, where does this leave us in our quest for understanding speciation, the smoking gun of evolution, you might ask? After exploring the long-term evolution experiment by Richard Lenski and Scott Minnick's refutation of Lenski's claim that he had observed speciation, we've landed back at square one. The question of speciation, the cornerstone of evolution, remains unanswered. The citrate-eating E. coli adaptation, initially seen as a beacon of speciation, has instead led to a death spiral of spectacular devolution, as was pointed out by Michael Behe. To make matters much worse for the unfinished business of Darwin's smoking gun, the study of ancient bacteria by J. William Schopf and the findings of the 2002 paper titled "The Paradox of the Ancient Bacterium" have further reinforced the paradox. They reveal a striking similarity between ancient and modern bacteria, both morphologically and at molecular levels, challenging the conventional understanding of speciation. As Schopp stated, modern pond scum differs little from the ancient billion-year-old blue-green bacteria. And as 2002 study found, almost without exception, bacteria isolated from ancient material, 250 million years old, have proven to closely resemble modern bacteria at both morphological and molecular levels. In short, although bacteria would be, because of the short generation times and large populations, idea candidates for speciation, there's a spectacular lack of change and/or lack of speciation in bacteria at both the morphological and genetic level for as far back in Earth's history as we are able to see. So the question of speciation, evolution's smoking gun, remains unresolved, and for all intents and purposes, indeed looks to be forever unresolvable for Darwinists.